I forgot you. I didn't listen to you, did I? I filled out the form now. All right, how about now? I just pulled the muscle. Okay, so one of his lesser, but to some of his very important contributions is this idea of evolution by sexual selection. And now I'll do my stupid British accent. The two characteristics affected by such a process, namely plumage development in the male and sexual preference for such development in the female, must thus advance together with ever-increasing speed. So in this is a capsule quote that any sexual selection researcher can recount, at least paraphrase, but what he's describing is what later became known as the runaway process, coevolution of a male trait and female preference for that trait in the face of making mating decisions. Well, Fisher, as you might tell from his glasses, which are pretty thick, had very poor eyesight. And so he could visualize a lot of mathematics and do solutions, and it was painful for him to write down models. He probably had a specific model in mind, or probably could have done it in an afternoon, but he never wrote down his model. And so there was a lot of debate about how you could produce this model. And Lande, in 1981, this is what, some decades afterwards, produced a formal model of the process and showed that indeed you could get this runaway, although for reasons we'll see it's probably implausible. And the basic idea in Lande's hands was that you've got a distribution of some male trait. I'm going to call it the ornament, just so you think of something like the peacock's tail, or the size of the peacock's tail. And it's normally distributed in the male population. Here's the average size of the size of the tail plumes in a male bird of paradise, the red bird of paradise, in fact. And then in red, we have a more subtle idea. This is the distribution of female preferences in the population. And what this curve means is that the modal value, this is the size of the male tail most preferred by females in the population. And it's slightly larger than the average, the modal size of tail in the population. So that's exerting a directional selection, moving the male distribution to the right. Pretty simple idea. But now specifying the equations for the coevolution of those distributions, bit of a tricky thing. And I'm not going to show you the details because they're in great detail in this publication I alluded to. But the upshot of this model is what I want to skip to now. And it looks like, well, let's look at the ingredients first. I forgot my slide order. Okay, so here's the ingredients in Lande's version of the Fisher-Lande process, and this is what we imagine Fisher had in mind. In the first place, there's a male ornament, say tail size, which affects male survival. We'll specify how. And via female preference, his mating success. Okay, so it has two effects. It has two effects on two different components of fitness. We also imagine there's female mating preference also with a sort of normal distribution based on the ornament. But it does not affect the survival or fecundity of the female. So if you think about that, that means that the preference is selectively neutral in this original Lande. We're going to relax that in a minute. If you freak out, if you're freaking out here, don't worry, we'll get rid of that. Secondly, that's a joke. Thirdly, both traits are heritable, and because of assortative mating and sexual selection on the males, genetically correlated. Raise your hand if that was completely understandable. Two geniuses in the room, excellent. But for the rest of us, the idea is this. Think back to that distribution. The females with the strongest preference in the population are mating with the males with the most extreme values of the ornament. And at the other end of the distribution, the same. Now at the high end of the distribution, the males that are most favored by the female are also leaving more progeny, right? So as a consequence of that, you build up a genetic linkage between the preference in the male sex and the ornament characteristics in the male. That is to say, males that are most preferred by the females produce sons with large ornament values, but they also produce daughters with extreme values for the preference. And that's a manifestation of this linkage that's built up and manifested and maintained in the population. And that's a key to some of what we're going to see in a minute. Okay, so now skipping to what happens with the process, there are two versions. One version is the version that Landy was talking about, the runaway. And you can show in the model that sometimes you can get specify the model so that you run away in two different directions, towards loss of the male trait or towards elaboration of the male trait, and that these trajectories, populations started here, will move away and evolve at ever increasing speed, or ever increasing speed towards loss. This runaway process, though, requires sort of implausibly large values for some of these genetic parameters. So I and people I've managed to convince think that this walk towards version, this sort of stable version of the model, is more probable, more plausible. And that in this version, populations wherever you start them move with ever decreasing speed towards a line of equilibrium, and then they stop when they reach that point. So this line of equilibrium then specifies the outcome. And if the population is of less than infinite size, finite size, then the populations, once they reach this line, will drift along it, and they'll be driven back towards the line. And that drift along the line then could produce an adaptive sexual radiation in the male ornament and the preference if we want. So what we want to do is characterize the process so we can make predictions about the kind of the characteristics, the statistics of an adaptive radiation reaching this line and moving along it then by drift. All right. So I'm going to sketch that model for you, and but first let's look at this troubling element. Not troubling, but there's a little tension. Once you're on that line I just showed, the line of equilibrium, you stop evolving because you've reached a balance between sexual selection red and natural selection in black. Now, what's natural selection? Well, think of a bell-shaped, not bell-shaped, but sort of parabola-shaped curve shown here in orange, and your fitness is a function of where you are on that line. So that if you reach a point here in the population, there's a net force of directional selection that's pushing you back towards this adaptive optimum under natural selection, right? That's what this curve is saying. And what we're imagining is that all along that line, that force of natural selection, this is called an Ornstein-Uhlenbeck process, back towards the optimum, is counterbalanced by this force of sexual selection in the opposite direction. And there are many different ways to strike this balance, and that's what forms this line of equilibrium. So let me say too, while we've got this up here, that we've standardized things here, so if the variance of this curve is 1, then the variance-like parameter for this curve is about 9. It's about 10 times broader. That's to say, this is relatively weak. This value for stabilizing selection is a value that's commonly observed in natural populations. It's not implausible. And we'll use that value later. So the OU, the Ornstein-Uhlenbeck, is like a rubber band, like that, constituting that black arrow, pulling the population back towards the optimum. We're imagining that that's happening on the male trait, and we're going to imagine that a completely analogous diagram could be produced for the female preferences. And I'm not even involving, except their curve would be red, but there wouldn't be any balancing force of sexual selection on the preference. Okay. Now I just added selection on the female preference, and so that's a generalization beyond what Landy was talking about. Okay. Now here, let's return to our problem. Here's our problem. We've got a model, but we need to connect it up to data. And right now, the basic problem is there's a lack of a connection in the literature between models of the kind I just described, even though there are about 30 versions with various elaborations. Nobody's ever connected them up with an adaptive radiation of anything. And I think it's connecting them up with a sexual radiation that makes the most sense and makes the most powerful test of the model. We haven't done this. So I'm going to sketch with the bird of paradise example how we could go about doing this. And so when we look at birds of paradise, now how many of you have thought about birds of paradise? How many of you have been to New Guinea? No, I haven't either. Have you? And did you see Birds of Paradise? Still, that's vastly better than anyone else in the room. So good for you. What's your name? Mike. Good for you. I've been in Indonesia, and the next island over the horizon was not New Guinea, but the northernmost Bird of Paradise island, but it was just over the horizon. But anyway, it's on my bucket list to go there. But how many of you have heard of Birds of Paradise? You know what I'm talking about? Yeah, right. So and their elaboration of plumes and all sorts of things is so extreme that they have the most extreme plumage in the bird world, and they are the most iconic example of evolution by sexual race. So they make a great test case. And so what we realized to use them was we needed these characteristics, and in general you need a time-calibrated phylogeny. You need a tree for the unfolding of the Birds of Paradise, and we need to know the time points for the nodes on that tree. We need to have male display that is obviously the focus of female choice, and as I'll show you in a video in a minute, that's pretty clearly the case with Birds of Paradise. And then most importantly, we need data on the components of the display, and I'm going to show you data on, length of wings and length of tail, and I'll show you why we want to use those characteristics. Let me show you these ingredients in more detail. Here's a mitochondrial DNA phylogeny of Birds of Paradise concocted by a Dutch group, Iristed et al. In 2009. And here's the Birds of Paradise. And basically what they are able to show by time calibrating their phylogeny is the common ancestor for the whole radiation of 30, 40 species is about 24 million years ago. And all of this diversification has unfolded on that tree in that period of time. Here's this red Bird of Paradise that we did in sort of diagrammatic view. Now, as I'll point out in a minute, we only have data. I could only find data. I should have gone to a museum like this one, and collected data. I didn't. The only data I could find in the literature was for this little group up here, and I'll come back to that little genus Paradisaea in a minute. So we're just going to do a test on a fraction of the phylogeny, but they have a representative manifestation of this larger craziness in plumage and call and every aspect of sexual communication. All right, so here's a picture of a Bird of Paradise, and this is a picture made by Tim Lamon in New Guinea, and it shows the birds displaying. But I think the best thing to do is for me to show you a video here, even if we have to sacrifice time in equations here. Do this. Whoops, go away. Go away. YouTube trying to close tabs, no close to it. Oh, jeez. This is harder than it looks. YouTube trying to take over my presentation.
Opportunity here to film all the phases of courtship, from calling to attract females all the way through to copulation.
I'll just leave it at that size.
Notice how that female's coming into the frame there, and it sends the males into this kind of round of excited galloping. So they go down one way and they kind of bounce back up, and they pause and they ruffle and shake their feathers. They look like they're cooperating, and they are displaying in a synchronous way, but really they're in competition. Both want to be the one that mates. And then eventually, after several rounds of that, they'll turn head down, holding themselves still so the females can see their vibrant yellow on the underside of their flank plumes. And when the females are interested in mating, they get in really close and inspect the males. And he starts doing a wing flapping that's more exaggerated. And he goes into this backwards dance. Back and forth, kind of hopping, doing this unusual thing. At this point, the female is actually allowing the male to make contact with her, albeit from his compromised head-down position. When he spins around then, and this is the pre-mating display, the male moves in and starts clapping her with his wings, giving this strange vocalization.
Do it. Do it yourself.
There's a lot of components of the display that do seem a bit aggressive. If I don't know that he's hurting the female in any way, but he is definitely clapping her with his wings, and he's simulating pecking at the back of her head and nape, even though he's not really pecking at it. The male will keep doing this for a while, and if the female is ready to mate, notice how she's kind of fluttering out her wings a little bit. That's the signal that she's ready to mate, and this guy is successful. Good morning to be a greater bird of paradise.
Indeed. Okay, did you see that final act there? That's the cloacal kiss. So that's the culmination of sexual communication over in a fraction of a second, well, a couple seconds. But there's a lot of sexual communication going on. So this is part of the display. There's almost no variation among living birds, much less among birds of paradise in that part of the display. And yet we're going to explain that fact with our model as well. Or we'll do that in the question and answer periods. As well as, so we want a model that explains that, the absolute, almost absolute, stasis in the cloacal kiss, and we want to go all the way to the other extreme and explain the incredible diversification in plumage and on all the multimodal things that go with it. So you noticed from that video that the takeaway is that this display is very, very complicated, and it's multimodal. It's calls, it's tactile, it's visual, and there's repetition, there's choreography. It's a very complicated thing. So to think we're going to model the whole bloody thing in evolutionary time is ridiculous, right? But I'm going to take just the simplest model and say, let's just pull two traits out so we're at least capturing the multivariate aspect. And as Dan knows, if you can do two traits, you can do three, you can do 100 in a vector. So just going from 1 to 2 is a big step. So that's all we're going to try and do, is just do a bivariate version of the model that would get us on the path towards explaining all that we want to understand about how the red and all the other birds of paradise came to be. Okay, so let's now talk about the model we're going to try. I've alluded to some of the characteristics in the model. We're going to do two male display traits, just a way station on the way to having a whole collection of ornaments in different sensory modalities. Two female preference, that is to say female mating is based on two traits, not just one. Selection on the female preference, it's going to be stabilizing like the OU curve that I showed you. We're going to let finite population size be finite. So really what we're talking about is a selection drift model to produce the radiation. And here's what it's going to look like. We're in tail length, wing length space. This blue curve represents the distribution, the bivariate distribution of wing length and tail length in the population. And then this is the bivariate distribution of female preferences for tail length and wing length. You can see that would exert selection in this direction. And we're going to show the bivariate mean of the males and the bivariate mean of the females as a dumbbell, just in some of the slides that I'm going to show you in a minute. All right, so questions? What? No, you can ask questions. Okay. Say what? Oh, yes, I do. And what we're going to imagine is that under selection and mutation, we've equilibrated a certain amount of genetic variation in both the traits and for the pairs and the pair of traits. So mutation's in there, but we're just going to set it as a constant parameter that's equilibrated with sort of average amount of selection on each of these characters. So I'm not going to talk about it. But believe me, it's right there in the appendix for the paper if you want to look at it. There's more to it than that. Okay, so there's good news and bad news about the model. The good news is the model's more general. We've got selection, it's multivariate, it's selection and drift. The bad news is that it's just complicated enough that I can't solve it analytically. And what I mean by that is I can't derive an expression that we know that the distribution of the trait and the preferences is going to be bivariate, well, 4-variable normal as a function if we take a time slice. I won't go into why, but we know that. But what we want is an expression that would tell us the variance of that 4-variable normal distribution, that 4 multivariate normal distribution at any point in time into the future as the radiation unravels. As replicate populations fill up 4-variate space. But even though I can't do that, and I haven't talked to anybody into helping me yet, we can determine the model's behavior with simulations. And so I'm going to show you what the model behavior looks like with a few simulations. But I can't spend a lot of time on it because we've got to get to the test. But let me tell you about the model. So here's the lineage mean for trait 1, ornament 1, lineage mean for trait 2, and here are a bunch of means in the population. And what you can't see is that there's an ellipse like this which represents the force of the sort of 95% contour of it's not a distribution, but treat it like a distribution. And then you can draw a 95% contour which represents, in this case, very strong natural selection on the male distribution. And here, weaker bivariate selection. Remember that orange curve that I showed you? This is the bivariate version for preferences. And it's weaker, and hence there's more of a dispersion at some point in time in the female means. This is what I mean by modeling the radiation. But here there's no connection. There's no sexual selection. There's no bearing on the male's evolution of the preferences. These are just two pairs of traits evolving independently. And so they will equilibrate then about their and buzz about the means about their respective adaptive peaks. If, on the other hand, we need to make selection on the preferences weaker, because if we make selection on the ornament weaker, then the traits go to the preference optimum, and we get an even smaller radiation than we'd expect under natural selection alone. Okay? So let's do that. Let's make selection on the preferences weaker. The preference peak pulls the ornament values in blue means over to that peak, and the preferences buzz about it. I did that fast. It'll make a little more sense if we look at some animations. And maybe I can even show them to you. I have to think about what I'm doing here. I have to escape and do this. Oh, darn. I shut them down somehow. Yeah, I try bringing them back up. I'm only going to do this, get one shot at this though. It takes a while for these to load, which is why I was trying to do it beforehand. But in my experience, you'd rather watch these animations than anything else I have to say, so except the video. Load for me, baby, load. All right, that's not too bad. Okay. Here's a caricature of the strength of bivariate natural selection on the male ornament. And here's the mean bouncing around the optimum at the center. And why is it not at the optimum? Because it tends to drift away because of finite population size. There are errors of parentage that cause the mean to drift away. And then that rubber band is pulling it back towards the optimum. The upshot is, you can see the ghost of positions past here. The upshot is you end up having this drift about this optimum. And if you had replicate lineages, as you do in this diagram, you could get, here I'm showing like 25 replicate lineages. You can see that it's the simulation starts and then quickly equilibrates, and you can see those 25 lineages buzzing about their respective optima. You're mesmerized, aren't you? Some of you are. Okay, let's go down to the sexual selection case. This is the one that interests us. Now we toggle in females exerting sexual selection on the preference. And what we end up with is the means buzzing about the preference optimum. And you can see I've connected them with a dumbbell. Each ornament mean is co-evolving with its preference mean. And you can see that I've actually started the population right at the preference optimum. If I started it over here, they'd quickly evolve to this position. But anyway, this is the sort of quasi-equilibrium that's rapidly achieved. So the point is, on the face of it, this seems like it might explain at least bivariate evolution in characteristics like wing and tail length, because the radiation is far bigger than the radiation you'd expect just under natural selection alone, right? The preferences are creating much more diversification in the amongst these 25 lineages than you expect just if we had natural selection. But the question is this enough to explain the birds of paradise? This is a radiation, but is it big enough across at equilibrium to explain what we actually see in the birds of paradise? I won't go into the issues of scale on this diagram, but even at this point, we can say it doesn't look like it's gonna be quite as powerful as we need. Let's see, what am I doing? Close current tab. Oh, I had another one open. All right. Okay, so now let's try and see if we've got an idea here. We have a model. We can simulate it. Can it explain this bird of paradise radiation? Okay, so here are the what is this? 7 species up at the tip. This is one genus, the genus Paradisaea, and for this genus we have they're monophyletic genus. We know the time scale for differentiation. We've got a tree over this 9 million years, and thanks to our colleague Mary LaCroix at the American Museum, we have data on wing length and tail length for, I've got multiple populations of some of those species, so there are actually, I think, 13 populations. Now, ignore this. Well, let's talk about this. These are the wing length and tail lengths for the males. And here I've got the 95% confidence ellipse, and these are the principal components, also known as the eigenvectors of that distribution, right? And here is here's the wing length and tail, not preferences in this diagram, but wing length and tail length, not preferences, but the actual analogs of these measurements. And you can see that the male diversification is considerably greater than in the females. Again, a sign of asexual radiation. But also notice that I have to tell you that I've standardized the traits so that within the population they have a standard deviation of 1, variance of 1. And we plotted things here so that we can measure the width of the differentiation in male wing length in units of phenotypic standard deviation. And whichever trait we look at, that width of the radiation is about 10 to 12 phenotypic standard deviations wide. Right? We're sampling from the end of a little phylogeny, a little radiation that's been spinning out there for, 9.3, 9.4 generations. Generation time of 5 years, that comes out to be about 1.8 million generations. Okay, so this tells us how much differentiation we expect as a function of time in these birds, at least out to the full length of the radiation, 1.8 million generations. So notice that before I showed you perfectly round clusters of means. If we want different shapes, we can do that by imposing, for example, correlational selection, that is selection favoring combinations of traits, and you can get, instead of round ellipses, you can get patterns of data like the one we were just looking at. All right. So the question is, can the model produce the pattern we're looking at on the time scale we've been given by the birds of paradise? And this figure summarizes the test. And so I have to take a minute to explain this to you. So here, this is the result of simulating 25 populations through time for 100,000 generations. We've set the width of the selection on the male preference at 9. That's like that diagram of the OU process I showed you. And we're just going to look at one ornament trait, right? I'm just going to back out of the bivariate case for the moment. Let's just do a test calibration of effect on the time scale we need. And what I'm showing you is that if we have these parameter combinations, strong preference, strong stabilizing selection on the ornament, very, very weak selection on the preference, an effective population size that's kind of small, 1,000, then if we look at the replicate lineage is evolving with these parameters over 100,000 generations, we see the populations evolving out to about 15, plus or minus 15 phenotypic standard deviations. So that's the amount of differentiation with this model that we see in the birds of paradise in about 1.8 million. We can produce the amount of differentiation among species and populations in 100,000 generations. We actually had 1.8 million generations to do it. So there's no question that the model can produce the results. And notice that without sexual selection, we would have equilibrated the ornament values inside this pair of black lines. We're way, way out there. And here's where we would have been. Here's natural selection on the preference. Both the preference and the ornament have been pulled way outside of those bounds by sexual selection. Now here's another interesting thing to notice. Look at this line right here. What you see here is the preference means evolving through time in this lineage, and here you see the ornament means in lockstep. And so this is sort of default what you get in the model. Over a broad range of parameter values, you get within a lineage the preference and trait means evolving in lockstep. So close that even without observing the preferences, you can infer where they are because they're always a tight ghost of the ornament itself. Right? So that's important because I forgot to mention, nobody has data on adaptive radiations and preferences. Just very hard to do. But what the model is telling us is, under this process, when you look at the evolution of ornaments, you're essentially looking at the evolution of preferences because they evolve so tightly. They co-evolve so tightly. How am I on time here? I'm almost, what have I got, 10 minutes? Or should I have stopped? You're waving me on. I've seen speakers now go on for a half an hour, but I'm not going to do that. Okay, but I have to pick up the pace. Okay, so we got the model to explain what we need to explain on the time scale. We need to explain it by making extremely weak, like a flat line model for preferences. The other way to do it is to have the optimum of the preferences move by some random process, say Brownian motion. And that's what I've assumed here. And so doing that so that this is the 95% confidence limits for where the optimum is under a Brownian motion. And then here are the lineages chasing the natural selection optimum for the preferences. And we can nicely explain the evolution of preferences and ornaments out to about what we need, and on the same time scale. And the amount of movement that we require in the optimum is just a small fraction of the standard deviation in an ornament per generation. It's just a little bit of movement, but accumulated over 100,000 generations, it easily produces the amount of differentiation we need. Okay, so that's good. All right, so here's where we are. Here is where we are. For one thing, evolving preferences control the evolution of ornaments. That's that tight tracking I just talked about. We can account for the diversification patterns in single and multiple, and the extent. We could do it either by assuming very weak stabilizing selection and relatively small effective population size, 1,000, 5,000. Or we can move the natural selection optimum to modest rate. So I tend to attracted to this one because it's a little easier to imagine what that means than extraordinarily weak, almost selectively neutral preferences. But we could talk about that, I think, at the end if you want. Okay, so here's the good news. Analytical solution. Well, now we're going to try the other approach, and that is, let's look at these generic models for adaptive radiation, stripping away the sexual selection specifics. Two big options out there, Brownian motion and Ornstein-Uhlenbeck, and I'm going to contrast them in a minute. The nice thing about these models is, although they don't address the sexual selection details, they are solvable analytically, so we don't have to simulate. We can just solve straight away. But the parameters we estimate aren't relatable to this model. They confound inheritance, selection, and population size. But we do account for the phylogeny, and we can use likelihood to compare the alternative models. The other good news is that we can, with a little bit of algebra, we can disentangle some of this confounding. So we can actually use them to estimate the models that we just had in this Fisher-Lande process. Okay, so here I'm going to show you the contrast between these two kinds of models. Then we'll test predictions with the bird of paradise with this program called Ouwie, and I'll tell you who composed it. And one of the things we're going to try and do is decide whether peak movement is a required explanation, and we're going to do that by using more than one selection regime in the parlance of this model, and that's like a poor man's version of a moving optimum. I'll show you what I mean in a minute. And then, as I alluded to just now, we can use this framework to estimate the parameters of the FLP, which we weren't able to do by the earlier approaches. Okay, here's what these two approaches look like. This is Brownian motion. It's either genetic drift of the mean or Brownian motion of the peak itself. And in either case, it's an ever-expanding distribution of lineage means. So I'm plotting lineage means. Here are the replicates. And these are our 95% confidence limits. They'll be normally distributed under the central limit theorem. Here we've got an effective size of 1, and this is genetic variance, not really that important to us. In contrast, the OU, that rubber band, always pulling the population mean back towards its optimum, it creates bounded evolution. And so the radiation is stuck at a value. It doesn't continually expand. So we're interested in whether we get support for one or the other of these types of processes. All right, they aren't? Is that the ordinate? What do you mean it's different? Oh yeah. And it wouldn't matter if we, yeah, so this one.
If you drew the one on the right and the scale of the one on the left, it would be pretty narrow down the middle of it.
Professor Gingrich is exactly right, and the scale is, so this is saying that this produces, with the same parameters, right, a very, very modest radiation, but also the pattern's completely different. So this one is problematic in fits because it tends to blow out. But in any case, let's put them to the test. This testing framework that I'm going to show you will test both kinds. The testing framework was devised by two guys named Ballou, working in Brian O'Meara's lab at Tennessee. And here's Brian. He teaches with me, so I had his picture at hand. And here's the phylogeny for our birds. I haven't shown that to you. And here it is, time calibrated at the nodes. And what we're going to say is, in this model, one of the options is, let's have a different value for the strength of stabilizing selection or genetic variance, whatever we want, in regime 2. And I've arbitrarily taken just this subset, with common ancestor here, and say, okay, suppose the regime shifted there, would that help us fit the model? For example, if the peak moved at that point, is that better than having a stationary peak? All right, so I'm going to flip through these slides quick. You can see them in the article, but basically what they show is that Brownian motion actually does a better job than any of the other models, than the other two models. So here's Brownian motion with a different value in regime 1 and regime 2, and here's OU with different values in regime 1 and 2. Two regimes are having two regime values for different values in the two regime does better, but Brownian motion does marginally better than this one. All right, so I don't think that tells us a whole lot, but let me show you what happens when we translate those parameters, which we can do here. If we translate those parameters using a little quantitative genetic theory, and what we can show is that this Brownian motion model here is equivalent to assuming in the drift of the mean case that we have an effective population size of 25,000 within populations versus 700 in the red. Now that seems biologically pretty wild. Birds of paradise populations ranging in one part of the phylogeny from 25,000 effective size to 700, kind of weird. On the other hand, under the other interpretation of variance of peak movement, both of those they're slightly different, but they're tiny, tiny values, both of them. So this is a very easy to imagine that this is a validation of the idea that the peak is moving at a very modest rate, but slightly faster out there in the red part of the phylogeny. And that does better than having a value that remains constant. Here it is down here in both parts of the tree. All right. I want to wrap up here without dwelling on this too much. So here's what it means to have a stabilizing selection function of 25,000. It's like a flat line. It's almost like saying preferences are, I've shown it with the blue distribution, but imagine it's red if you want. Okay, so here's the conclusion. Brownian motion actually fits best, and it's the most plausible model for the data. And its conclusions are consistent with the phenotypic Tango model. And in particular, we end up concluding that something with a moving optimum probably is the best model. And if that optimum is moving stochastically by Brownian motion, that gets the job done. Okay, so where do we want to go with this? For one thing, this probably doesn't bother you, but it does bother me to some extent. I'd like an analytical solution for this model that Lynn and I concocted. And that includes a model that includes peak movement, because that's sort of an add-on right now. This isn't a very big data set, and it would be nice to do a test like this with some other radiation where there's more data. And then finally, if you'll notice that what I did is I fit the model, estimated parameters, and then looked at the parameters and say, are those biologically reasonable? Well, another way to proceed would be to use a Bayesian framework in which you bring in prior information about heritabilities and population sizes in birds, fold that in, and then estimate the parameters at the end, and all in one big exercise and package. Now, my colleagues Weta and Harmon have done essentially that, but I should use their program and see what results it comes up with, whether it estimates the parameters in adequate way. It needs a little tweaking though, because this model is bivariate and they've sort of got a univariate model. All right, acknowledgements. I've got a group of people, women and men, that I teach with in the summer. With Joe Felsenstein as the co-organizer, and we call our course Evolutionary Quantitative Genetics. And it was teaching this course with Brian O'Meara. Where's Brian? He's in there somewhere. Anyway, it was teaching the course. Sitting back, I realized, oh, I could be using Brian's technique on my own data. So the idea for this talk was sort of hatched in this working group. And here are my co-instructors, and here you see the students as well. It was sponsored by Nesset and Nimbios, but now both have folded and are in the process of folding. And we've moved the course to the University of Washington in San Juan Island, the Friday Harbor Labs. We taught it there last summer, and we'll teach us this summer. I'm bringing this up because here's Brian acknowledged, but I haven't found him yet. If you think you like this kind of thing, you could take this week-long course in June. And if that's of interest to you, you need to Google Evolutionary Quantitative Genetics 2018, because the applications close on March 1st, or see me. And that's it. Thank you very much. Yeah, okay, all right. Maybe I'll get the guy behind you, Dan. You've got a, yeah. Yeah. They don't rule that out. Good genes isn't in this model, but good genes is sort of a genetic consequence rather than that's not really selection on the preference per se. So selection on the preference would be on the order of saying, if you're a very, very finicky female who wants a male way out on the tail of the distribution, you might spend so much time looking, waiting for that male, that you incur an enhanced chance of predation on the lek waiting for him to appear. That kind of selection. And when we say weak, we're saying, but that's a very, very, you know, like your mortality rate goes up by, you know, like a, you know, I don't know, something out at the 5th decimal place. That's what we're talking about. So this testing approach that I've talked about could fold in lots of things like good genes, like maternal effects, all sorts of additional things. So what we did is strip the model back to the really core original model, 81, make it bivariate, make it finite populations, and try it with it. And even then, we couldn't solve the equations. We had to do a simulation. But if you wanted to try and add handicaps or some other wrinkle, you could do this. You could do that with the same framework. We just haven't gone there yet. Dan?
So, if we're explaining the bird of paradise correlation, it seems like any model that has general processes as part of the model has to explain why most radiations don't do what birds of paradise do. Do you have any intuition for what it is that makes this different, the radiation of birds of paradise different? Because most avian radiations obviously do not do what birds of paradise do. So, is there something that,
Yeah.
what is your thinking on that?
So it's almost trivially easy to tighten it up, and all you do is make selection on the preferences. So the evolution of the preferences is the key to the process. As preferences evolve, so do the ornament. So all you do is tighten up the selection on the preferences as in that flip, like in that flip version. If you make selection on the preferences very tight and selection on the ornament very tight, you can have complete stasis. You can explain the stasis of the cloacal kiss, right? That element in the same courtship. So in that sense, I think it's a general model in that you can go all the way from plumage elaboration to complete stasis in the final stage of sexual communication, just by changing. Actually, one key parameter, well, two. Tightness of selection on the preference and how much that preference optimum moves. You can come up and talk to me afterwards if you want.
All right, Damien, take you, please.
Thank you.